Hello everybody. This is the second tutorial on how to do the HHMI Annul lab, which we'll be doing today. There are different ways of getting to it. I will share the link along with this video, but I'm just going to go straight through Google here. And it should take me to the right spot. Now, I've had trouble finding it on the website itself, which is why I'm leaving this up to Google to find for me. This is the one that I want. If I did save some data previously, I want to sign back in to Google. I'm using my PHS account for that. And it should say that everything that I need is here. What I want to look at today, the ecomorphs, I really just wanted to get some data on practicing taking measurements. I want to compare these measurements. You might by now have noticed some of the data. Something like the A. sheplandi would be a twig annul, so would A. occultus be a twig annul. These live on different islands. This is the island where Haiti and the Dominican Republic is, while this one is Puerto Rico. A. evermannii is one of those canopy lizards, as is A. celestinus. Sorry, I haven't been practicing these names as much as I should. We have some trunk ground annuls. We have probably grass annuls. I would know that by the lines. If they're clinging to grass, that would help them blend in. This part we've completed. We've done the x-rays of the limbs. I did not have you do the toe pads. Not really that necessary for right now. I want to focus on limbs because that's where we've seen rapid evolution over a short period of time on different islands off the coast of the Bahamas. We're going to go today to Module 2, Phylogeny. Phylogeny is the relatedness of different species. There is a tutorial video here. What we're going to be looking at are DNA sequences. You may recognize these names of the different species of annul. This is one of those trunk ground annuls. This is one of those canopy lizards, lives up in the trees. This is one of the twig annuls that live on different islands. Now, what you'll remember, this is the actual data, again, that Dr. Lassos generated. This is made from a gene, a piece of DNA on the mitochondria. If you remember, mitochondrial DNA is evidence of bacteria being free-living prior to becoming mitochondria. What we're going to see is we have these long A, T, G, A, G, C, C. You remember those letters from the genetic code? What we're going to do is comparing these genes. This is one small gene on the mitochondria. We're going to be comparing these genes with this particular tool. So I'm just going to go Command-C since I have a Mac. I'm going to go to another website where I can actually conduct this search. I'm going to post all of my data here. This is a pretty sophisticated computer program that will allow us to compare DNA sequences on this gene. From there, if you remember, that over time we have small mutations in DNA. And some of those DNA mutations are silent mutations, which means that those 3 letter codes for the amino acids, maybe one letter gets switched to another, but it doesn't actually change the amino acid. Those silent mutations build up over time so that when we see these species compare their genes, we might see that certain genes are different in terms of their lettering. The more differences you have, the more mutations have built up, and the longer time it has been since these two species diverged from a common ancestor. Now, this may take a few moments, but what I want you to do is copy these sequences, put them in. It's trying to work to align these it'll work to build a tree of relatedness in these species. And you will be able to download a file that you will submit as part of your assignment for today. It depends on how... Okay, so here we go. What each of these lines does is it branches back to where these two species diverged from a common ancestor. You'll see that these two are closely related. The next closest relative to them would be this one. And if you're really checking, you'll see later on when we do some analysis, these actually come from the same area, the same island, in fact. These species diverged from the same ancestor more recently. This one diverged a much longer time ago. And this one isn't even an anole. It doesn't belong to the Anolis genus, which is the name for all of these different anoles. This is another kind of lizard, which means that diverged the longest distance away. Now, what I would want you to submit to me is a PDF, which you can download. Please put your last name up on this file. So mine would be Eastburn. And then you will share that with me. And let me go back to where I want it to be. From there, that's the next step. You did the sequencing. You probably got something that looked like this. If you remember these names, how they grouped, fairly similar to what we saw on my PDF. Let me see if I can actually show that to you. The PDF that I had is there. Let's see how my PDF compares. Sheplandi at the top, Cyboides, Occultus, Cristatellus. All of these are just names. The computer program that's analyzing this just sees that as a file name. It's not really making any... It's not establishing any knowledge on these names based on anything besides a name they were given and a DNA sequence that they were given. But we do see that they're all lining up based on what Dr. Lassos' data show, which is pretty amazing, pretty interesting. And from there, we're looking, as I told you, two species that connect closer to the edge are more closely related than one that connects farther back. According to the phylogenetic, phylogeny is relatedness, genetics is with genes, DNA. According to the phylogenetic tree in this virtual lab, anole lizards are more closely related to one another than they are to Leocephalus carinatus. We have to go through some questions. Anolis cristatellus is more closely related to Anolis occultus than it is to Anolis evermanni. So I have to find Anolis cristatellus. I have to find Anolis occultus. I have to find Anolis evermanni. If I notice, these two go back farther than the first connection, but they don't go nearly as far back as with occultus. So cristatellus is more closely related. That's actually false. Now that you have familiarized yourself with the tree, let's try to detect some patterns. Here's where you're going to go. For example, the twig anoles, you're going to link all of them together, all the T's. There's two of them. Two trunk crown anoles, which are at the top of the tree. Two of the trunk ground anoles, which are at the bottom of the tree. Trunk ground. And two of the grass bush, which live in the grasses. They generally have they generally have those lines on them. I'll go back to the full screen so you don't see any more text from my daughter. Based on this phylogeny, do lizards of the same body type tend to be more closely related to one another than lizards of a different body type? That's absolutely false. We have the two twig anoles, which are not each other's closest relatives. We have the two canopy anoles, the ones at the top of the tree, that are not each other's closest relatives. They were also comparing species from two different islands. This is the island with Haiti and the Dominican Republic, and then we have Puerto Rico. Now, what you might expect to see here is if they live on the same island, maybe they came from a common ancestor more recently than the ones on different islands. And that's exactly what we end up seeing in this particular case. All of the Puerto Rican lizards are more closely related to each other than they are to the Haitian and Dominican lizards. What you're seeing also is that the lizards on Hispaniola all branched from a common ancestor more recently. Now, this is a little bit confusing here because we do see that the one lizard from Puerto Rico does group with two lizards from Hispaniola. These are all old branches, which aren't necessarily quite as reliable as newer branches. What this could mean is that there's an older population of lizards on Hispaniola, and then one of those lizards from Hispaniola came to be the lizard that was the founder population for Puerto Rico. That's most likely what happened in that case. In general, lizards on the same island, they're more closely related to one another. That is absolutely true. There's some questions that I don't think I can skip over. Same types of anole lizards live well. I will let you answer these and want to move on to the next part, which is really the last part that I would expect you to do today. This leads you through the experiment that Professor Lassos did, where he took these two lizards and took them to different islands that had been cleaned or washed clean of their lizards during hurricanes. And we start to see the difference in lizards over time. You can see a video on how they collected the data, which is pretty interesting. And we're going to measure the x-rays. Now, this is the part where I actually want you to do all of the measurements. It's a little bit tedious, but you'll actually be able to look at some data once we're done. We're going to measure the hind limb length. This is something that you did before. And we're doing hind limb. Whoops. That was clear last. Record the measurement. And body length. Right to the back of the hips. We're going to do this is with the lizards on the bigger island where there are still trees, the big trees where these ground trunk anoles don't need to have the shorter legs to grip the branches. So I want you to go through all of these, make your measurements, then go results, graphs. Now, unfortunately, I can't do this until I record all the data. So what I'll do is I'm going to fly through this. I don't want to make this a ridiculously long video. I will make the measurements and then I'll tune back in once I've made all of my measurements. By this point, I've done all of my measurements on Iron K, which is the larger island with the trees, where we would expect the lizards to have long legs so they can run from the bottom of the tree to catch their food and then run back to the tree. Now we go to the experimental island where we're going to do the same thing. Now remember, this was where you had a very small island with only bushes with small twigs possibly some grasses growing on it. So we should see if these lizards have adapted, we should see some variation or some difference in their limb proportions, which is what Professor Lasso saw. Now, again, I'm not going to do all of these on screen, but I did want to make sure that you keep going. You click here, you do hind limb length. You click here, you do body length, and then we keep going. I will pause this again until I get through all of my measurements. Just so you know, this may seem a little tedious, but this is how science works. We have to do very careful experiments with very careful attention to detail. This is why science works so beautifully, though. Once we understand and establish a procedure and we collect data in an accurate and reliable way, we're able to make conclusions based on the data that we collect, and they stand the test of time. They stand up through rigorous testing if they're well performed and the hypothesis is proved, which will eventually become a theory. Now I've completed both of these groups of lizards and done all of their measurements. I do have a data table here. This is what you should be able to generate. Now here, if we look, we see they divide the hind limb length by the body length. When you make that division, you find out how long the hind limbs, those back legs, are compared to the body. Here you see that the hind legs are a little bit shorter compared to the body than they are for the ones that live on the island with the trees where they would need to run faster. Here they're needing to grip branches. I'm going to save and continue because that's already been calculated. Whoops. I have to do the last ones here. So 18.7 for my measurement. Your measurement might be different. So make sure you check what your measurement is. 42.5, and that is 0.44. And over here we have 19.1 divided by 45.1. 0.4235. Notice how all of these numbers are rounded off. When you get to chemistry, you're going to talk a lot about significant digits. There really should be 3 significant digits here, but since the other ones are only giving you 2, 4, 2, I'll put a 3, but it won't let me put it in. Save and continue. This is now the measurement, the ratio of the back legs to the body length. This is the ratio right here. We're taking a look at the sample data. Standard deviation, we did a little bit with that at the very beginning of the year. Standard deviation is one measure to tell us how wide of a distribution of the data we have. Meaning, do we have a lot of numbers that are clustered in here? We'd have a low standard deviation. Do we have numbers spread out over a long distance along this curve? Then we have a high standard deviation. The more standard deviation you have, the less reliable your data in general. Okay, now that I've read this, I can see that we're going to calculate the means. My suggestion here is to open a Google Sheets. And now we're going to manipulate the data. I should be able to copy this. Command C, Command V, yes. Okay, that I don't need. What I can do here is insert, so down here I'm going to insert function. I'm going to do average. and this is from B1 to B10. 0.449. And then I'm going to get my other data. You know what? I do want to label my data. So I'm going to go lizard. This is on iron K. And this is on experimental island. Here I can do average experimental island. I'll copy the data again. Command C. Take it over here. I didn't get one of these. 0.42. I don't need this. I'll get the average again. I will insert the function. This is now from F3 to F11. Good. And then for standard deviation, same thing. I want to look for statistical standard deviation, STDEV. Standard deviation will be from now, my whole data set is shifted down one because I entered in the title. So it's going to be from B2 to B11. And that is my standard deviation. Didn't like that. I put it in the wrong location. I'm going to go back to statistical, standard deviation. We're going to do again B2 to B11. Here I want to call this standard deviation. And please be sure to share this spreadsheet with me. Last thing here, I have standard deviation. And go to statistical, STDEV. Now I'm at F3, 2 with the colon, F12. No, F11. And I get my standard deviations. These I'll now need to go back and put into my original data. 0.449. And my standard deviation is 0.0237. These numbers are fairly small. This one's a little bit bigger as far as the standard deviation than this one, which is also interesting because you have more variation with the lizards that live on the larger island. You have less variation on the smaller island, which actually would make sense. 0.0237. Let's try that. Okay. It will round off a little bit for me. And then 0.437. For my sample mean, and standard deviation 0.0171. Save and continue. You know what? Seeing how far and how long this is taking us, as long as we calculate sample mean and standard deviation, and you get that into a spreadsheet that you share with, we'll call this lizard spreadsheet. We'll call it lizard sheet. And you save it with Mark Eastburn at pps.princetonk12.org. As long as you get this sheet done, I think that's more than enough to get completed for today. So let me see your data, let me see your averages calculated, let me see your standard deviation from the measurements of these lizards, as well as the phylogenetic tree that you developed, the PDF that looks like this. Let me see those two things, and we'll call it a day. I think that's more than enough work to get done for one day. Thank you.